Hello friends, today we are going to discuss about extranuclear inheritance or cytoplasmic inheritance. We already know that in the nucleus, genes are present in the chromosome known as the nuclear genes, and these are responsible for the inheritance of most of the traits from one generation to another. So, they play a very important role in inheritance, but these are not the exclusive source of inheritance. Certain experiments were performed and these suggested that in addition to nuclear genes, there are also genes present in the cytoplasm, both in prokaryotes and eukaryotes, and these are known as the extranuclear or cytoplasmic genes. And these are also responsible for inheritance of certain traits. So, if we talk about eukaryotic genes, these are of two types: nuclear genes, cytoplasmic genes. Nuclear gene shows the Mendelian pattern of inheritance and segregation. Cytoplasmic genes do not show the Mendelian pattern of inheritance and segregation. Inheritance of nuclear genes are known as nuclear inheritance or non-cytoplasmic inheritance. But inheritance of cytoplasmic genes is known as cytoplasmic or non-nuclear inheritance. Result of reciprocal crosses in case of nuclear genes are same. That is, if we take two variety of any plant, plant 1 and plant 2. If we take female gamete from plant 1 and male gamete from plant 2, then the progeny will be the same if we take it vice versa, that is, female gamete from plant 2 and male gamete from plant 1. So, the result of reciprocal crosses are same. But in case of cytoplasmic genes, result of reciprocal crosses are different, on the basis of which we can tell that nuclear gene shows biparental contribution and both the parents contribute equally to any trait or character. But cytoplasmic genes shows uniparental contribution, either paternal or maternal. These are mostly maternal. So, now what we can tell that eukaryotic cells possess main complement of chromosome in the nucleus known as the nuclear genes, and extra DNA molecules or chromosome is present in mitochondria, chloroplast, and in some cases endosymbionts. What are endosymbionts? These are the organisms that lives within the body or cells of another organism, and both are benefited from each other, or there is mutual benefit among each other. Example: kappa particles, sigma particles. If we talk about prokaryotes, example in E. coli, then these possess a single main chromosome and it is not present in membrane bound organelle, it is known as nucleoid, plus these have extra DNA elements that is present in the cytoplasm and it is known as plasmids in case of E. coli. So, now let us talk about characteristics of cytoplasmic inheritance. Cytoplasmic inheritance lack Mendelian segregation and typical Mendelian ratios. Characters that is inherited by cytoplasmic genes persist over many generations. And in case of the reciprocal crosses, different phenotypes of progeny are obtained. These shows uniparental inheritance of trait, either maternal or paternal, but mostly it is maternal. These does not show linkage to nuclear linkage groups and assort independently. So, now what is the definition of cytoplasmic inheritance? It is the inheritance of cytoplasmically located independent self-replicating nucleic acids, and these differ from the chromosomal genes by their location within the cell and have their own nucleotide sequences. The smallest heritable extrachromosomal unit is known as plasma, genes, and all of the plasma genes of the cell constitute the plasmin. So in case of the cytoplasmic inheritance, cytoplasmic genes that are present in the cytoplasm is responsible for the inheritance, and it is independent, it is self-replicating nucleic acid, and these differs from the chromosomal genes in their location, because chromosomal genes are present in the nucleus, but their location is in the cytoplasm. Extranuclear inheritance by cellular organelles. It was first described by Carl Correns in 1908, and this inheritance was by chloroplast, because DNA molecules are present in chloroplast. These chloroplasts are present in the cytoplasm of the cell, and hence this type of the inheritance is known as extranuclear inheritance or cytoplasmic inheritance. And he did an experiment in 4 o'clock plant, scientific name Mirabilis jalapa. He studied on the variegated variety of the Mirabilis jalapa. In the different branches, different types of the leaves are present. In some branches only green leaves are present. In some branches white leaves are present, and in some branches variegated leaves are present, which shows both green and white patches or mosaic of both the green and white. So what he showed, that if the seeds that is obtained from the green branch, these give green progeny irrespective of whether it is pollinated from the pollens coming from white branch, green branch or variegated branch. Similarly, the seeds that is obtained from the white branch give only white progeny irrespective of any type of the branches from which the pollen grains is coming. In the same way, seeds obtained from the variegated branches give all the 3 types of the progeny: green, white and variegated, irrespective of the branches from which the pollen grains are coming and pollinating the female gamete or egg. Let us see this in detail. Phenotype of the egg bearing branch. If the phenotype of the egg bearing branch is white, this is the phenotype of the pollen bearing branch, and if it is pollinated from the pollens coming from the white branch or the green branch or the variegated branch, the phenotype of the progeny is always white. Similarly, if the phenotype of the egg bearing branch is green and it is pollinated from the pollen coming from any type of the branch, white, green or variegated, then the phenotype of the progeny is green. And if the phenotype of the egg bearing branch is variegated and if it is pollinated from the pollens coming from any branch, white, green or variegated, the phenotype of the progeny will be all 3 types: variegated, green or white, because cytoplasm present in the egg of the variegated branch contain all the 3 types of the chloroplast or plastid: white, green and variegated. So what we can conclude, that the phenotype of the progeny always resembles the female parent. These are the female parent, and progeny always resemble the female parent. If it's white, then white; if green, then green; and if variegated, then all the 3 types: variegated, green or white. So the genes that are concerned or that are responsible for the color of the leaves are located in chloroplast, and this chloroplast is present in the cytoplasm. These are not present in the nucleus. So it is cytoplasmic inheritance. And here egg contributes the cytoplasm along with the chloroplast, and pollen grains contains very little cytoplasm, and these are mostly devoid of chloroplast. Hence, without chloroplast, pollen cannot affect the phenotype of the offspring. Therefore, female gamete or the egg is responsible for the cytoplasmic inheritance, because mostly the cytoplasm is contributed by the egg, which also contain the chloroplast, and hence in this case it is known as the maternal inheritance. Since the chloroplast its own DNA and it is present in the cytoplasm. It is also known as cytoplasmic inheritance. So we are going to discuss about extranuclear inheritance by mitochondria. Here we will discuss about the inheritance pattern of peptide mutants. Like chloroplast, mitochondria also consist of its own DNA, and this mitochondria also present in cytoplasm, so it is also known as cytoplasmic inheritance. So what are these peptides? Peptides are mutant. It was first discovered in yeast Saccharomyces cerevisiae, that is also known as the baker's yeast. In the yeast cells, normally what we see, large colonies when these are grown in the agar plate, and these are known as granae. But in rare cases, small or minute colonies are also seen. These are known as peptides. This is due to the slow growth rate. Here, the number of the cells are fewer compared to the cells present in the large colonies, but the size of the cell is same. And why these have the slow growth rate? Because this type of the cells consist of mitochondria which has defective electron transport protein. And how the mitochondria has defective electron protein? This is caused by the mutations in mitochondrial DNA. And since the mitochondrial DNA is present in mitochondria, which itself is present in cytoplasm, this type of the peptide is known as cytoplasmic peptide. There are also certain peptides which results in the mutations in nuclear encoded genes, which are involved in the oxidative phosphorylation. Hence, these are not cytoplasmic peptides. Inheritance of these peptides was described by Boris Efrasi et al. What he observed, that there are three types of the peptides: segregational peptide, neutral peptide, and suppressive peptide. Let us see their inheritance pattern one by one. Inheritance of segregational peptide. Here the peptide cell, which is haploid, is crossed with the normal cell, which is wild type or grande. This is also haploid, and when these are crossed, which this gives the diploid zygote, which is normal. Then the zygotic meiosis takes place, both meiosis 1 and meiosis 2, and we get the peptide and normal ascospores in the ratio 2:2. So this ratio indicates that there is Mendelian segregation taking place. So what we can conclude, that segregational peptide follow Mendelian segregation. So these are controlled by the nuclear genes, or in the segregational peptides, the defective mitochondria is due to the mutation in the nuclear genes, because this nuclear genes only shows the Mendelian pattern of segregation. But there are certain peptides that are also extrachromosomal or non-segregational. These are also known as the vegetative peptides, and the genetic basis of these peptides is the Rho factor. This Rho factor is the cytoplasmic factor, and when this Rho factor is absent or defective, then it results in the peptides. So in this type of the extrachromosomal or cytoplasmic peptides, or in the vegetative peptides, this Rho factor is absent or defective. One such peptide is neutral peptide. It is shown by Rho 0. That means it completely lacks the Rho factor. Let us see its inheritance pattern. When the haploid peptide is crossed with the haploid normal, then it gets diploid zygote, and this is normal. Then this undergoes zygotic meiosis, and the progeny or the spores or the haploid cells which we get are all normal. So on the basis of this inheritance pattern, it was explained that neutral peptide lack most or all the mitochondrial DNA. So most of the genes are lacking which encodes for the electron transport protein, which is involved in oxidative phosphorylation, and hence this is the cause of the defective mitochondria, which results in neutral peptide. So when this neutral peptide is crossed with the wild type, then the cytoplasm of the wild type is the source of the normal mitochondrial DNA in the resulting progeny spore, because cytoplasm of the wild type has the normal mitochondria, hence normal mitochondrial DNA. So the progeny which we get are all normal. Therefore, what we can say that neutral peptides are not transmitted. Now the next is suppressive peptide, which is represented by rho minus. This has the defective rho factor. Let us see its inheritance pattern. When the haploid peptide is crossed with the haploid normal, we get the diploid zygote, which is peptide. And when this zygote undergoes zygotic meiosis, then we get the progeny, which are all peptide. So here also it shows the non-Mendelian pattern of inheritance. Suppressive peptides contain mitochondrial DNA, which is greatly altered in base composition compared to the wild mitochondrial DNA. Hence, these suppressive peptides contain mutated mitochondrial DNA, while the normal ones contain the normal mitochondrial DNA, which is also known as wild type. So the progeny which we get as a result of the cross between suppressive peptide and normal haploid cell are all peptide. There are two hypotheses to explain this result. Mutant mitochondrial DNA that is present in the suppressive peptide replicates more rapidly compared to the wild mitochondrial DNA in the zygote, as a result of which most of the progeny are peptides. And the second hypothesis is, in the zygote, recombination occurs between mutant and wild type mitochondrial DNA. This introduces errors into normal mitochondrial DNA, and now most of the mitochondrial DNA are also mutated, as a result of which most of the progeny or the offspring that we get are peptide. This is all for today's video. If you like this video, please hit the like button, share it and subscribe my channel. Thank you.